Excitation-contraction coupling is the process by which an action potential generated in a motor neuron triggers acetylcholine release at the neuromuscular junction, which produces an endplate potential that initiates a muscle action potential. This action potential will spread down T-tubules to activate specific dihydropyridine receptors that mechanically gate ryanodine receptors on the muscle cell's sarcoplasmic reticulum. That causes the release of calcium, which binds to troponin C to initiate cross-bridge cycling and muscle contraction. In this tutorial, I'm going to review the neuromuscular junction and discuss how the action potential generates that acetylcholine release to produce the endplate potential. Next, I'll discuss how this process is impacted in various muscle disorders. I'm Dr. Catherine Moore, the Histology Wizard. First, let's use this simple diagram to review the components in a basic neuromuscular junction. We'll start with this structure on the left representing that presynaptic terminus of the motor neuron, and on the right, we see the postsynaptic structure, the motor end plate of the muscle fiber. Between the two, we have an area called the synaptic cleft, a tiny extracellular gap between the presynaptic terminal and the postsynaptic membrane. It's really small, it's only about 50 to 70 nanometers. Now, essentially, this is the space that the neurotransmitter, in this case acetylcholine, has to cross after being released. Now, the cleft is not just empty space, it contains a structured basal lamina that helps organize the pre- and postsynaptic machinery, and it also anchors an important enzyme in place. So the synaptic cleft contains the enzyme acetylcholinesterase, which rapidly breaks down acetylcholine after it's bound to its receptors, terminating the signal. So this enzyme is critical for terminating neuromuscular transmission, meaning it rapidly hydrolyzes acetylcholine in the synaptic cleft into choline and acetate. So that prevents prolonged activation of the postsynaptic receptors, and it allows the endplate potential to repolarize so the muscle can relax. It's also how we regenerate acetylcholine. Without functional acetylcholinesterase, acetylcholine would simply accumulate in the cleft, and it would continually stimulate this receptor, producing a depolarization block. So the muscle would contract initially, but then become unresponsive over time. Let's look at a few more key players. On the presynaptic side, we find PQ type, or CAV 2.1, voltage-gated calcium channels. These are concentrated at active zones, specialized presynaptic membrane regions where synaptic vesicles dock and fuse. Now, for convenience sake, I'm just kind of showing them a little bit far away from the vesicles. But essentially, when calcium enters, it does so in very close proximity to these docked vesicles, and that creates a high local calcium microdomain, so a high concentration that efficiently triggers protein complex assembly that's critical for vesicle fusion, without requiring calcium to diffuse so far. Now next, we have calcium, seen here in blue. The calcium is going to flow in from the extracellular space through those presynaptic PQ channels when they open in response to depolarization from the arriving action potential. So this is different than many other calcium signaling processes in the body where intracellular calcium stores are important, such as in skeletal muscle contraction where calcium is stored in that sarcoplasmic reticulum. Now up next we see synaptic vesicles. These are small membrane-bound packages that store acetylcholine, and they cluster near those active zones. Not shown here is a special transporter called vesicular acetylcholine transporter, or VCHAT, which pumps acetylcholine into the vesicles for storage. Next, these red circles indicate neurotransmitter, that acetylcholine that's going to be released from those vesicles and bind to receptors on the postsynaptic side. Now, we've already talked about the cleft and neurotransmitters, but I wanted to touch on a set of cellular machinery that's critical for this neurotransmitter release, called the snare complex. So the snare complex is a family of proteins involved in vesicle fusion. It's named for soluble NSF attachment protein receptors, but the name doesn't really matter. Importantly, there are three proteins that work together in this complex: SNAP25 on the presynaptic membrane, syntaxin on the presynaptic membrane, and synaptobrevin, all sometimes called VAMP, on the vesicle membrane. These proteins are going to work together to facilitate vesicle docking, binding, and release of neurotransmitter. Now, when this complex is disrupted, for example, if any of these proteins is cleaved, this snare complex can't assemble properly and vesicle fusion and neurotransmitter release can't occur. All right, let's now head on over to the postsynaptic motor endplate, where we find nicotinic acetylcholine receptors, or the NM receptor. These are the primary target of acetylcholine at the neuromuscular junction. They are ligand-gated ion channels. And when acetylcholine molecules bind, those channels are going to open and allow sodium influx, generating that end plate potential that triggers muscle contraction. All right, we've got our components for the first couple of steps in excitation-contraction coupling. Let's put it all together to see how it works. So we're going to start with an action potential traveling down the motor neuron axon, which reaches the presynaptic terminal. Depolarization of the terminal membrane is going to open those PQ-type voltage-gated calcium channels concentrated in active zones. Calcium will flood in from the extracellular space, create a high local calcium microdomain around those synaptic vesicles, and that calcium influx triggers assembly of that snare complex we just talked about. So synaptobrevin on the vesicle zippers together with syntaxin and SNAP25, pulling the vesicle membrane into fusion with the terminal membrane and releasing acetylcholine into the synaptic cleft. Now, this is shown in slow motion here, but this happens in microseconds. Now, acetylcholine is going to diffuse across the cleft rapidly. And I've not shown this here, but for the nicotinic acetylcholine receptor to open, you actually have to have two acetylcholine molecules binding simultaneously. That's going to cause a conformational change that opens the central ion channel. That's going to allow sodium to rush in and a bit of potassium to flow out, which depolarizes the motor end plate and generates that end plate potential. Now, importantly, the end plate potential isn't really an action potential itself. It's a graded depolarization of that end plate region, and if it's large enough, it spreads to the surrounding sarcolemma or muscle plasma membrane and triggers a full-blown muscle action potential via voltage-gated sodium channels. Meanwhile, we have acetylcholinesterase that's anchored in the cleft basal lamina, and it's going to rapidly hydrolyze that acetylcholine into choline and acetate. So choline will be taken back up into the presynaptic terminal by a high-affinity choline transporter and recycled to synthesize new acetylcholine via choline acetyltransferase, or CHAT. All right, now that we understand the key steps required for successful neuromuscular transmission, from calcium influx at the presynaptic terminal to acetylcholine binding at the postsynaptic receptor, let's look at how disruption of some of these steps produces muscle weakness and impacts function. Several important conditions and toxins each target different points in this pathway, so understanding their mechanisms is important for understanding diagnoses. We're going to first start with myasthenia gravis. Myasthenia gravis is an autoimmune condition in which antibodies, most commonly directed against that postsynaptic nicotinic acetylcholine receptor, block and accelerate degradation of these receptors, which reduces the end plate potential below the threshold necessary to trigger a muscle action potential. Now, because each successive nerve stimulus depletes the available acetylcholine and there are fewer functional receptors to compensate, patients experience a hallmark feature of fatigable weakness, which means symptoms that worsen with repetitive use and improve with rest. Next up, curare. Curare and its derivatives are clinically important as surgical muscle relaxants, and historically, curare was used as an arrow poison to immobilize prey. Curare and its derivative non-polarizing neuromuscular blocking agents, such as rocuronium and vecuronium, work by competitively blocking that postsynaptic acetylcholine receptor. They bind to the same alpha subunits as acetylcholine, but they don't activate the channel, thereby preventing depolarization of the motor end plate and producing a flaccid paralysis. Now, unlike myasthenia gravis, this blockade is competitive and reversible, meaning we can overcome it by increasing synaptic acetylcholine concentrations. which is exactly the mechanism behind reversal agents like neostigmine, which inhibit acetylcholinesterase and allow acetylcholine to accumulate and competitively displace curare from the receptor. Next, we have Lambert-Eaton myasthenic syndrome, an autoimmune condition in which antibodies target the presynaptic PQ-type voltage-gated calcium channels, and that reduces calcium influx into the nerve terminal. And this impairs the snare-mediated vesicle fusion and acetylcholine release. Now here the problem is presynaptic. It's related to insufficient calcium accumulation, so repeated stimulation allows calcium to build up progressively at the terminal. And that results in a characteristic incremental response or transient improvement in muscle strength with repeated use or high-frequency nerve stimulation. This is exactly the opposite of what we see in myasthenia gravis, because in that case, the problem is downstream of acetylcholine release. Now, Lambert-Eaton is associated with small cell lung cancer in around 50 to 60% of the cases, where the tumor itself is expressing those PQ-type calcium channels, and that triggers this autoimmune response. So Lambert-Eaton is considered a classic paraneoplastic syndrome. Finally, let's talk about botulism toxin. Botulism toxin is produced by the gram-positive anaerobic rod Clostridium botulinum, and it acts on the presynaptic terminal by entering the nerve terminal via endocytosis. It permanently prevents vesicle fusion and acetylcholine release by binding to the snare complex. And that prevents vesicle fusion at the presynaptic terminal and ultimately blocks the release of acetylcholine. But let's dig in a bit deeper to see how this happens. So after entering the nerve terminal via endocytosis, botulism toxin releases a light chain, a zinc-dependent endopeptidase that cleaves snare proteins, specifically synaptobrevin, syntaxin, or SNAP25. That totally depends on the toxin serotype. Unlike Lambert-Eaton syndrome, where calcium influx is impaired but the machinery is intact, in botulism, the fusion machinery is itself destroyed. So no amount of calcium accumulation or repeated stimulation can restore neurotransmission. This produces a flaccid, descending paralysis that does not improve with repetitive nerve stimulation. Clinically, botulism presents in several forms, including foodborne, wound, and infant botulism. The latter occurs when the botulinum spores, classically from honey but not always, colonize the immature infant gut. They germinate and produce the toxin in vivo, and that can explain the presentation of a hypotonic infant with autonomic features such as dry mouth, sluggish pupils, and constipation. Now let's finish by putting this all together. Myasthenia gravis and curare affect transmission at the postsynaptic motor endplate. Lambert-Eaton prevents calcium influx leading to lack of vesicle fusion, and botulism toxin impairs the vesicle fusion process itself. Each of these produces characteristic muscle weakness or paralysis, and understanding the pathophysiology is critical for understanding diagnosis of muscle myopathies. Thanks for stopping by.